Foraging for food in a wild forest is a high-risk activity. Pick the wrong cap, and a single bite can result in severe illness, organ failure, or a deadly outcome. This complexity is mapped out in identification flowcharts, showing how lethal varieties display standard, innocent traits. A mushroom might have white gills, a subtle ring, or a bulbous base, features occurring in both safe and toxic species, leaving little room for error. Despite that risk, the nutritional profile of edible varieties makes them a unique food source. Some mushrooms contain 19 to 35% protein by dry weight, alongside high concentrations of vitamin D and essential minerals like selenium and potassium. These concentrated nutrients are why fungi are integrated into medicinal frameworks, like the Ayurvedic system shown here. Because they provide specific antioxidants and immunity-boosting compounds, certain mushroom varieties are processed into traditional treatments for managing blood pressure and chronic respiratory conditions. Because the nutritional and medicinal benefits are so high, but wild foraging remains dangerous, humans had to develop methods to bypass the forest floor and produce these organisms in a controlled, predictable environment. Success in cultivation requires a shift in how we view the organism. Mushrooms are not plants. They lack chlorophyll and cannot produce food from sunlight. Instead, they survive as saprotrophs, meaning they grow by breaking down and absorbing nutrients directly from decaying organic matter like wood. This diagram shows the anatomy we typically recognize: the cap, or Pileus, and the stalk, or stipe. But this is only the temporary reproductive fruiting body. The actual body is the mycelium, a permanent network of microscopic white threads living entirely underground. To farm mushrooms, a cultivator does not plant seeds. Instead, they must create an environment that allows this mycelial network to thrive, protect it from competing bacteria, and eventually trigger it to produce its fruit. That fruiting process begins under the cap. These thin vertical slits, called gills, act as a reproductive factory. They are lined with specialized cells that produce microscopic spores. Maturing gills release millions of spores. Once a spore lands, it germinates into a primary hypha. Two compatible hyphae must fuse, forming the secondary mycelium. This network bundles into a hyphal knot, which expands into a pinhead, eventually maturing into a full mushroom. By identifying these specific biological stages, from spore release to mycelial fusion, cultivators learned how to replicate these conditions indoors to trigger growth on a massive scale. Modern mushroom farming looks more like an industrial laboratory than a farm. It takes place in dark, moisture-controlled environments where every variable is managed. The process begins with substrate preparation. Cultivators boil materials like hay or straw for about 30 minutes to kill off germs and mold. This creates a sterile, nutrient-rich food source for the mushroom. Spores are then introduced into these sterile bags of hay. Over several weeks in total darkness, the mycelium colonizes the bag, turning the straw into a solid white mass. By introducing fresh air and water, the farmer signals the mycelium to begin fruiting. This method allows farmers to take the natural, unpredictable life cycle of a fungus and concentrate it into a highly efficient production line inside a plastic bag. Once harvested, the challenge shifts to preservation. Because mushrooms have high moisture content and no protective skin, they spoil faster than almost any other crop. To prevent spoilage, farmers use various preservation techniques. Short-term storage relies on refrigeration, while longer-term methods include pickling in brine or using chemicals like potassium bisulfite to stop bacterial growth. Traditional methods, like the process for making sukuti shown here, involve washing and thinly slicing the mushrooms before drying them in the sun or an oven. Removing the water content allows the mushrooms to be stored for years without losing their nutritional value. These agricultural and preservation systems mean we no longer have to depend on the forest floor. We have replaced the life-threatening uncertainty of wild foraging with a stable, scientific process. By understanding the specific requirements of the fungal life cycle, humanity transformed a high-risk gamble into a sustainable global industry that provides safe, nutrient-dense food